the PDF. So that PDF, basically the material that I'm showing you today, and then also here you will find the links to all the notebooks and different things that I will show in the live demo later on. Okay, so let's go back to the presentation. If I manage, there we go. All right, so first I'm a scientific officer at the Center for Cellular Imaging. We are located at core facilities in Gothenburg, and we're a national facility that integrates both light and electron microscopy. We offer open access to all the imaging equipment, and this level of access depends, of course, a little bit on your project and what is your experience. But our standard way of working is that we will train you in using the microscopy equipment, so you can be independent as soon as possible. We are welcome to researchers from universities and companies in Sweden and abroad, and we are also part of Bioimaging. So people coming, from abroad, then they might enter or apply for service via the Bioimaging website. Now, the center is a very, let's say, multidimensional, in the meaning like we have many different microscopes that can give you different types of contrast from your sample. And then usually what we would like is that we would like to take a single image and actually move it through the different equipment so we can benefit from the advantages of each one of them, right? And then depending how we prepare the sample, in some cases, we actually manage to do what we call these correlative workflows, where we actually take exactly the same, piece of tissue or cells or sample, and we move it through the microscopes. So you might get images that have different resolution scales, different contrasting agents, and different kind of methods to create contrast. So then, of course, there is a thing that joins them all together, which is the one at the bottom right, which is the image analysis. And that's a little bit what we're going to be talking about today, right? So first of all, so why image analysis, bioimage analysis in this case have become so important is because more and more right now we're moving into what we call image-based quantitative biology, right? So as if you are a biologist, you know that a lot of your assays are already quantitative. You have many types of measurements that you might do in your lab. And in the past, imaging used to be more for like phenotypes. You will put your cells in the microscope, look at them, and then, for example, see if you recognize one phenotype versus the other. But now we're moving away from that and we're moving into more quantitative assays. And then kind of the thing that we buy in when we go into quantitative microscopy is that we believe that each image has very much a lot of rich information about the underlying biology. So what is happening into the cells can be assessed by the way they look. Therefore, we should be able to extract descriptors from our images. So we need to translate that image into some sort of table of descriptors. And by following those descriptors and comparing, for example, different conditions or different drug treatments and so on, we should then be able to make conclusions as to what is happening at the biology level, right? So what you see here at the bottom is a classical example of one very nice article that shows this idea. What you have is that you have cells where they have created basically a healthy kind of cell line, and then they have the disease model on the right side. And then what you will basically see is that the images that I show there on the left side, they look distinctly different to you by eye, right? And then if you have a situation like this, you could come up with an assay where you say, okay, now what I'm going to do is I'm going to take my disease cells, I'm going to treat them with a bunch of different, medicines, or perhaps at different concentrations in different conditions. And I'm going to see if I can recover the disease-looking cell into something that looks like a healthy cell, right? And if you just do this for a few conditions, you can check by eye. But if you do this for thousands of compounds, then this is not possible anymore. So now, what is going to be our biggest challenge is the fact that biology is very complex and we have a lot of variability, right? So a cell is not just a very, like a constant organism. It has its own cell cycles, but even within that, we will have also cell differentiation, and we will have, of course, different type of models. So there is a lot of variability, and somehow, depending on how common the phenotype that we're looking after occurs in our system, we might have to actually expand longer and longer the libraries, because we need to be able to catch that very not common event that occurs in, within thousands of cells, right? So this is basically one of those things, is that we are gonna have to handle rare events. So our pipelines usually will have some sort of baseline, and what we're aiming after is finding those rare events and trying to understand why they happen. So one of the ways in which in microscopy we have kind of tackled this problem of quantitative biology is what is called high content screening microscopy. Is just some sort of automated microscopy. There is different levels of automation and different levels of screenings, right? So how many cells and how many plates and so on. But basically the idea is that if I go here at on the left side, what you have is that you have your model, and then you do some sort of perturbation. It could be either drugs or genetic perturbations or whatever. And then you put all of those cells in a multi-well plate, okay? It's just basically a bunch of little, you know, for those that are not biologists, just a bunch of little, you know, you could think rooms where you put your cells. And then you're going to go with your automated microscope, which I try to describe here with this objective at the right side. And you're going to look at each one of those rooms and take a picture of the cells. And you're going to look at how are my cells looking like, depending on the different conditions that I know are present on each one, right? So in some screenings already on the right side, you will see that already from the macro scale, just looking at the full well, you might get some information. Maybe you are just looking at how many cells are alive versus dead and things like that. And in principle, you don't need super high resolution for doing something like that, right? But actually the level of resolution then can be changed depending on the objectives that we use. And then the type of descriptors that we can describe are completely different, right? So we could look at the well level, we could look at the population level, like the second image that I show, or we can even go down to the cell level, okay? So depending on what type of biology we're looking after, we might have to change the experiment a little bit. All right, so once we have those images, we have all of those conditions, and we have all of these tables of descriptors, then is when the bioinformaticians usually jump in, because now they have humongous data tables, and now they are capable of mining all of that information to try to explain the biology that is behind, all right? So the purpose of today is mainly try to describe how we go all the way to the data tables. I'm not gonna go into data mining, all right? But I'm just trying to show, so my purpose here is as well that perhaps we can try to bridge a little bit the different communities, so the communities of the biologists and the, you know, we have the image analysts like me, but also we have all the bioinformaticians that are very good at doing all the data mining. And perhaps this could be a way of introducing the topic and that people get to know what type of images we have, what type of data we have in microscopy. Now, this automated microscopy also opens a very, very cool method, which we are right now investing at the facility, which is called smart microscopy. So what I have shown you before is kind of this sort of box on the right, which is that we could have very big computers looking at our images in very complicated ways, and try to do different tasks such as segmentation, translation, or restoration, and so on and so forth, right? But those images have already been acquired, okay? You have a humongous data set of images, you process them, and you get numbers. Now, what we are trying to do is that, as I described a little bit before, we will also have automated microscopes, right? So I can use a computer to send instructions to my microscope to describe what experiment I want. Now, what we can try to do is that we can try to put them all within the same bucket. So we can do everything at the same time. So the idea is that the microscope is going to look at the sample, it's going to take a particular image, we're going to do some image processing, and then we can figure out, for example, is these cells in the right conditions to do a higher resolution screen? Yes or no? They are in the right condition. Okay, now I send a message to my microscope that says, please change the behavior, right? I'm not going to talk too much about it today, but there is one of the things that we use a lot here is actually Python. Because there is a very cool project called Micromanager that actually allows you to control your microscopes and is very open. So if you have all your image analysis already in Python, then you can use Micromanager to sort of put things together, right? So if you're interested in this type of things, also please let me know later on. So now, for the topic of today, bioimage analysis and Python. So the question is, okay, why are we doing these sort of things in Python nowadays? So first, what is bioimage analysis? So bioimage analysis, you could describe it as the process of identifying spatiotemporal distribution of biological components in images, okay? And then the idea is that we want to measure these characteristics in an unbiased way, okay? So we want to basically have a set of rules, and we agree on those rules, and then we look at all the images with the same rules, and then those rules are made in such a way that we can try to understand underlying mechanisms of biology that are happening via the images, okay? A classical pipeline can look something like this. So we have an image loading step, and then there's a lot of things that could happen in there. Those include data conversion, metadata handling, and proprietary file formats. If you do microscopy, you know that this is the land of the file formats. You know, each microscope has its own, and then we need tools to be able to open these different ones. There's a lot of work being done already for that, but also at keeping the metadata, because we might want to know, okay, how this image was acquired, how long was the exposure, so on and so forth. Okay. And once you have your image and you have your metadata, then you start with the pre-processing. This is going to be things such as filtering your images, doing some kind of stitching. If you imagine now that you have a very, very large field of view that you want to cover, but your microscope can only look at a small place, then you have to take several images and then you have to stitch them together, for example, and you might have to do corrections due to uneven illumination or something along those lines. Once you have a nice pre-processed image, then we go into segmentation, which tends to also involve something that are called morphological operations. So I don't know if you are aware of these things, but once you segment an image, it basically becomes a binary mask, right? So basically you can have a particular number that designates, okay, 1's for my cell of interest, 0 for my background. And then you might do some operations to clean up those segmentations, perhaps remove small objects or things like that. Once everything is clean, then you can start to quantify. And then those ones we do things such as connected component analysis, object filtering, and so on and so forth. All right. So the idea is that here on the top, you see a little bit of that example. You see raw data, filtered data, segmented data, then a table of all the objects, and then finally a little number that describes, for example, the area of the cells. All right. So now there is another thing which I think is important, especially in this community, is to keep an idea of there is some differences between what we do and what people usually classically identify as image analysis or computer vision in other fields. Because in many other fields, what they are trying to do is that they are trying to replicate, human accuracy at certain tasks, right? And object detection as a human would do them. There is a lot of things that we borrow from that field, but what we want to do is that we do not emphasize human recognition. What we emphasize is objectivity while doing the measurements. That's the part that is most important for us. Okay, so that brings sometimes some long conversations over BS as to how you should do things or not, right? Just because of that small change in mind. Now here we go to the thing that actually defines the field. I think in many ways is that the bioimage analysis is a humongous landscape of many tools. Because what tends to happen is that each laboratory have created a very good tool for a very specific task, right? And then what, I try to show you, this is a forum where people that do bioimage analysis go and discuss and ask questions, and you will see there is something that I call the community partners, and then you see all the people, right, that has a particular software that is very good at doing a particular thing. They are there involved, and you can see how long the list is, right? Now, if we try to look into those, then there is some examples of programming languages that are a little bit more common than others. But in principle, there's a lot of things that is developed in the Java world. Those include, for example, ImageJ or Fiji. Then we're going to have Python. And that, of course, is very huge in all the machine learning community. And then we're going to have things such as MATLAB as well and some other software, right? So the idea is that, unfortunately, because of this fragmentation, what tends to happen is image analysis pipeline that I just showed you before, that it has some processing, segmentation, and so on, different steps. What you tend to do is that you tend to choose a software that is good for that particular task. So usually you will do one task in Fiji, then you will do another task in Elastic, then you do another task in MATLAB, and you do another task in another software, right? And this was very common, right? So what tends to happen is that then you end up sometimes with a bit of troubles because you want to, you have to handle all that pipeline. It's very difficult to describe to other people. It's very difficult to share, and so on and so forth. Now, what the thing with Python is this, what I like to call the one ring effect, right? So one software to control them all. So the idea is that in principle, there is something which is attractive about Python, which is that despite its learning curve, there is packages for many different things. So you could stay in Python for all of the steps if you want to. Okay, so that's one of the kind of the selling points about Python, is that you could try to do all of the things within the same language. So then you have only one learning curve to go through. And also, if your pipeline is already described and explained, you can share it much easier because it's only one thing to go. Now, there is a lot of things about Python that we like. So it's in terms like, let's say, easy to learn, depends how you look, from which perspective, but from a programming language perspective, it's relatively easy to learn. It has a lot of ways in which we can share packages, and it has a thriving community, right? So that's very, very attractive. Now, it's also, I think, very good because now people are getting more and more used to this concept of the Jupyter notebooks, which is what I'm going to show today. And they're very good for training, and they're also very good for sharing what you have done. And of course, the new thing or the new kid in the block is all the machine learning stuff, right? So a lot of the machine learning world lives in Python. So if you want to use that for bioimage analysis and you already know Python, you have a huge advantage, all right? Another thing that happened, and I will demo that today, is Napari. Napari is an image viewer for microscopy data in Python. And it's a project that has received a lot of funding, especially from the CZI, from the Chang Zuckerberg Initiative. So the other reason why I think Python is moving so strongly is because even companies, or let's say money, which is not classical research money, is being injected into these projects, right? And that actually is giving a lot of energy to the community, and we, people are very thankful, of course, for that collaboration. Now, this is a little bit of a repetition of what we did, what I showed before. But I think that what I wanted to concentrate a little bit was on the challenges now. So I think that the challenge that we have if we move to Python is generally the teaching to the beginners, because many of these tools, we want biologists to be able to use them, because not every pipeline is extremely complicated. So they should be able to do simple tasks very fast and quick. And that's what ImageJ or Fiji is great at. You can just install it and start playing immediately. So, and this is really what the community is trying to do with Napari. So Napari now has a way in which you can install packages and you can run them directly from the viewer. So it gives you a feeling of Fiji. So that's one of the things that we're moving forward. And the other thing is that, of course, there is, I think, a lack of general knowledge in some fields about programming languages, especially, something like Python. So I think there is a lot of things that we can do also from the education side, that if we just teach a little bit of programming to people, this will become a lot easier, because they don't need a specific programming, we should not have to teach bioimage analysis. It's not the same as teaching programming. So if we manage that people know a little bit of programming, then we can really concentrate on the bioimage analysis. Now, the very exciting things or the new opportunities is that we have access to a lot of other communities that live in Python, especially the machine learning, and now the hardware control and the smart microscopy. So I really think if you start to see the trend right now, a lot of people are moving there. So if you are just starting in bioimage analysis, I would recommend that you invest the time learning this and not just stay only with something like Fiji or ImageJ. This is a slide that I borrowed from a friend, just because another thing which I really like about this is I said a thriving community, right? So so it happens that you start to do bioimage analysis and you realize that you actually like programming, then you are in the right crowd if you are there, because there's a lot of different projects where you can get involved from, even if you're a beginner or if you're extremely advanced, you can jump in and start to participate. So if you like this type of collaboration, this is the place to go. So there is a lot of community-driven projects and also openly extensive ones. So with that, I'm gonna move into a short demo. But before that, perhaps, I don't know if there's any questions either from you guys or from the chat that I can have a look in general of why would I spend my time learning Python to do bioimage analysis, right? Nothing?
No, nobody has written anything in the chat. Maybe we will take questions as things go along, if somebody thinks of a question.
All right, so if somebody has a question, just write it in the chat and then we take it, right? So we're gonna go into the short demo, and then my idea today is just to show a little bit how we actually do it in practice, and also to pinpoint people to a few websites and a few packages that would be like your starting point, right? So if you know these packages, you can start playing immediately. And after that, it's just a matter of exploring which packages are more applicable to your particular application or your particular question, okay? So first of all, it's gonna be Napari, which is the image viewer that I'm going to use. The other one is SKImage, which is gonna be the one for the processing steps, like the filtering and the segmentation and so forth. And then I'm gonna show a quick example of what happens if you have a proprietary file format. So you don't have just TIFF. how do you do it? And then I'm just going to show one with the CZI format, which is the one from Carl Zeiss microscopes. Okay. Again, all the information is available and it's there in the GitHub page. So let me try to slowly move there. So on the website, there is a very extensive README file. And I think another thing I'm going to do is I'm going to take this and I'm going to try to put it on the chat. Did that work? Yes. All right, so there is an extensive README file. I wrote this like if you were a student that has no idea about what Python is. So I hope that you can follow along. But if you are an expert, you can just skip to the very much bottom of this documentation, okay? So what I'm going to do is that as a prerequisite, you need to know how to install Conda environments. Usually, I'm also going to be using Git command line tools, although it's not completely necessary. And you can use any IDE that you prefer, but I'm going to be showing VS Code, okay? So then also you will see that this repository has an environment file. So you can just immediately use Conda to just reproduce my environment and start working from there. I tested this on 2 computers yesterday, but you know, things can happen. So if something breaks, just please let me know and I will help out. But I think you should be able to follow this also afterwards. I think there's a recording of the session, so you can also have it. Let me see if there's something important that I need to cover before that. No, I think that we're going to continue to our use case. So what I have done is that I have a copy of that repository with me. I'm using VS Code. And then it's a little bit annoying, this Zoom thing, but I think I can go around it. And the first script that I have is an example. There's no way I can move this, right? The top one is always there. I don't remember how. Well, it doesn't matter, I will survive. Sorry for that. So if it looks clunky, it's because there's a part of my screen that I don't have access to. All right, so what you will see is that I might have to actually go out from the full view of, how do I do that now? Sorry, let me stop my share for 1 second. There should be a way. Yeah, there we go. Up, so I can move things a little bit. Yes, and then I will go back to Zoom, share my screen, desktop share. Okay, now at least I can see everything. All right, so the first example is just something that we're going to use to run Napari from a Python script. Okay, so if you don't want to use notebooks, you don't have to. You can call Napari from also a script, and that's basically what we're doing. I'm also from SK Image, I'm importing just some example data, right? And I'm importing Napari. So what you see is that I create a viewer via this method, napari.viewer, and I'm putting data that comes from SK Image, right? And the other thing that I'm saying, for example, is I'm saying this is an RGB image. The way in which we can handle RGB images is slightly special because we don't want to modify the relationship of the red, green, and the blue, right? We want to keep them all together. So it always looks the same way. And then you basically start with the Napari run. So now we tested this before, I do promise, and it did work 20 minutes ago. So let's run it and hope for the best. And let's see if Zoom doesn't obliterate my computer. Hello. There we go. Okay, so that is Napari. Okay, so what you see right now is that this is the image, that's the RGB image, okay? And now you see that because it's an RGB, I don't have access to a lot of things. The color map is RGB, you know, it's settled. And then you see that I have some gamma correction, contrast limits, and opacity. And this is also the same, like if you have used some other viewers before, this is based on this concept of layers. So these are my layers. I can, you know, remove a layer or look at a layer and so on. I can delete a layer, etc. Now, Napari also, of course, we can zoom in and out very quickly. We can move along, and it's very responsive. But perhaps this is not the most exciting image. We also have other things such as, where is it? Hello, This one is not working. Interesting. Doesn't matter. So you can you actually can load a lot of different images, and that's what I'm going to show. So this is just a very minimal example of how you can just load one image directly from Python. Okay. Then we're gonna go into this one, and that one is, as you have seen, the Napari viewer is its own little UI, so it cannot live inside of a Jupyter notebook. So what I do is that I initially import Napari, but I also import this thing which is called screenshot, so I can create views. And then, for example, let's just run this code. So I'm going to create a completely new viewer, and I'm going to take a screenshot of that one, which should be empty. And again, my computer is running it very slow today. I assume it's Zoom, but we will see if it survives. Please survive.
You got a question in the chat. Alexander here, Google.
Yeah.
When you share your script, do you also share YAML file so you could use the content?
Yes. So yes, that is true. So it's very important, these environment files. If you are in the business of sharing with others, then you want to keep track of the things that you have installed in your YAML file. There are ways in which you can sort of ask Conda to do it for you, but sometimes there's a lot of documentation online. So yes, you have to keep track. Also, it's very important to keep track of versions. Conda in principle is very good at making sure that there will be no fights between the different libraries, but you never know. So it's better that you keep that as well under control. And another thing that I'm going to suggest is that if you are going to invest on a pipeline that you do many times, do not take, let's say, packages that are not being supported right now. So just look first before you invest your time that this package has activity, that people are maintaining that package, that that has releases that are not so far away. Sometimes it's better to build your own function, even if it takes you longer time, but you are building upon packages that are very robust, than trying to borrow something that a PhD student in Italy did 3 years ago, but it's not being maintained. Okay. All right, so this is working. Then the next thing that I'm going to do is that I'm going to import the input and output functionality of SK image. And once we do that, and I don't know if I will run all the cells because I see my computer is really suffering. So what you see, because I have run this before, I will go like this. So what you see here is that now I can take that input and output functionality to read TIFF files. Okay. Now there is other formats which are very general, and you can open with this. And I will show just after this an example of what happens if you have a file format which is not so common. Okay. Then, for example, I can have my viewer. I can add that image, and you will see that I can immediately give names to that particular layer. So what you will see, which I find that the people from Napari did really nicely, is that all the little buttons that are in the UI correspond as well to all the properties that you call here in the methods. Okay, so they did that very nicely. So if you go here, you will see that now my layer is called rice. Okay, you will also see that there is color map, blending, interpolation. All of these things you can set up when you're loading your image. Okay, and it's very easy because all you have to do, if you don't know how they're called, either you go to the documentation. That for a beginner that can be a bit problematic. But if you are just a beginner, just look at these names and you have a very good starting point. It's probably just called like that. Okay, so now I have this rice, and then the other thing that I'm going to show you is that Napari has a very nice way of handling different types of images. Now I'm gonna load another image, which is called a labeled image. Okay, so what that actually means, if I go into Napari, you will see now that each piece of rice has a different color. And if I go into here, you see that I have rice and I have labels. And here on the right, it has a little sort of label mark. It knows that it's a label layer. And that actually can be used later on during all our analysis, because now we know, okay, these are the images, those are the labels, and then I can do analysis of this image based on my level, on my labels, okay? And if I hover over, you will see that here on if you look here on the bottom left of the viewer, if I hover over, it says the pixel position together with the index. It says right now index 51. If I move to the next one, it says index 42. You see, so it's kind of, it's very interesting. And another thing that it has is that it has this prompt that you can use to interact with your layers. But I do know that for some reason that I still don't quite understand, when I call the viewer from a notebook, it is not responsive. So if you do it via Python and you have your viewer open, you can actually interact with your layers from here. all the same things that I'm doing in my code via my notebook, I can do directly here. So if you are a guy that loves command prompts, you already have it built in. Okay. I am more of a notebook guy. So, okay, this is just, again, just a very, very simple example. All right. So what I'm going to do is that for the moment, I'm going to close. And then I'm going to ask again if somebody else has any other questions. Yes, go ahead.
Can you get the function for exported from
Yes. Yeah, you can all the layers that you have, you can also export. And there is a lot of, you can just use the normal functionality of Napari, and that is usually like TIFFs. But you also have a lot of plugins that I will show the website at the very end that you can start to export into different type of file formats. All right. So all this exporting is possible. And also there is a lot of very nice, because Napari is a real 3D or multi-dimensional viewer. So you can have 3D and time, which is kind of nasty in Fiji. But Napari was really built for that. So it immediately out of the box is very good at 3D and time. So you can also do very nice videos and exports of videos of exploring data sets in 3D and time or different things. Okay. All right. So just in case that people didn't ask, there was a question here from the if you could not listen, because I think the audio is connected to my machine. There was a question about how to export images, and yes, you can export images from the viewer.
You said that construct a function call that corresponds to all the UI.
Yes.
Elements, right?
Yes.
And then I guess when you set the UI element to some value, can you also get?
Yes, you can get them.
A function call that has all of that already put in.
Yes, yes, you can. So you will see here, this one, I'm just gonna hover over for the, just in case that people are very beginner, but for those that are a little bit more advanced, perhaps it's interesting. So that's why I show it here immediately. We can come back to this example if we have time or if we want in the discussion. So for example, in here, what you see, what I call advanced load and view is the following. So what I'm going to do is actually, I'm gonna use NumPy to do a lot of operations on these images before I put them back into Napari. Okay, so that's a very common way that I work, is that I usually don't use Napari all the time. I use it to explore a data set. Then I do a bunch of operations. And then, you know, after all those transformations, I might want to explore it again. But also, for example, things as matplotlib or even skimage have very basic image plotting, right? So you can just look at your images as you work with them. So for example, here I'm using NumPy, and I'm using this library to be able to work with these CZI files. If you want to know which library is good for your particular file format, I would recommend just go to the forum or go to the plugins website. Then, for example, here, I'm going to say, okay, this is my path, and this is my CZI file. And interesting now is that because I'm doing it this way, I can have access to the metadata without having to load the whole file. Okay, so for very big files, this is very useful, because you might realize how large your data is before you load, and you might want to load just a specific part of your image and not everything. All right. Then, because I want to be constantly using not Napari, but matplotlib, for example, for my plotting, I did this helper function, so you can borrow it, which I just do is that I normalize my image into 8 bits in such a way that I can just constantly plot them, you know. So this is just a very, very much little helper function. So for example, here what I'm showing is that, okay, now that I have this one, I can read from my image file a particular channel or a particular set plane or a particular time point. I don't have to load the whole thing, which might be too big, okay? And the data that I'm showing today is what we call multispectral data. So I have 32 color channels, right? So I don't, if I load them all at the same time, it almost becomes very difficult to follow, like what's going on there, okay? So for example, I can say, okay, I just want to read channel 13, and I want to plot that image. And for example, here I see, okay, channel 13 contains a lot of information about the mitochondria, but also some information about the nucleus. Okay, this is just for me to explain a little bit what we see in the image. Then what I do is that this one, again, this is just me that has created a helper function for the loading, just to be able to do this in an easier way. Because now what I have to do is that I know that I have a lot of channels, right? So what I'm going to do is I'm going to loop over the channels, and I'm going to load all of this data into a NumPy array. The interesting thing is that NumPy arrays are completely compatible with Napari. So I can bring any humongous NumPy array and I can load it into Napari, and it will just work with it as its images. All right. Now, the important thing for Napari is to know what your axis in your multidimensional array mean. Okay, so when you load, you can actually choose which axis is the X dimension, which axis is the Y, which axis are the channels, you see? So everybody can, it's like a free-for-all. There is some recommendations, but you can also identify in Napari, okay, what is what, and it will know how to digest that. So for example, here again, I'm just using that function. I'm loading that thing. I recognize that it has 32 channels. This is how to create an RGB image from that, because if not, it's very complex and difficult to see. So I'm going to collapse the 32 channels into 3, and we're gonna. So let's try to see if we can run this guy, so it becomes a bit more interesting. If my computer has to be, yeah. So this again. So I'm reading the metadata. This is just to normalize my images, as I explained, and then this next cell is gonna just plot a particular channel. So you see that I didn't have to load the whole thing. This image is just an array of X and Y. And then this is to load the full data set, which could take a little bit of time, but this data is very small because it's for a demo. So now it says, okay, you have 32 color channels and you have 150 pixels by 150 pixels. This is just to change it to an RGB. And then here, for example, you see I'm using Matplotlib, okay? Because all I want to do is to just take a quick image of that if I want to keep processing, all right? So I don't have to go immediately into Napari if I don't want to. But now that I have built this representation with this is an RGB representation, I can then now decide, okay, I want to explore this better, and I can decide to go into Napari. So I can create a new Napari viewer, which is what my computer doesn't like when I'm on Zoom, apparently. All the other stuff works. Usually takes like 3 seconds.
Yeah, you had another comment in the chat. One that Alexander said, please not use the spider IDE.
All right, good to know. Thank you. I haven't tried that one.
It's the one that comes with that.
Yes, that's correct. Yeah. So okay, so now I have an empty view. If I check the shape of that image that I have created, as expected, now I have, you see X, Y, and now my channels are at the very end. So I have to be careful with that. So then, for example, I can extract each image. And then I can add, you see here, what I'm doing is that now that I have extracted each one of them, I'm adding them independently and I'm setting that color map. So I'm not handling this as an RGB image, as I told you before, because if not, I don't have access to change as I want the red, the green, and the blue. So now if I do this, okay, so here is a screenshot, but also we can see it live. What you see now is that it's a little bit boring because my layers are just on top of each other. So I don't get this feeling of an RGB image, right? And the reason for that is that because we have this blending here, okay, and that blending is not correct. What I want them is to be sort of semi-transparent, okay? So then what I can do is that here I can check what was that property. So I can say, hey, viewer, the blue layer, please tell me what is your blending? And then I might have to say, no, I don't want this one. I want the additive one. So again, I can do it via the command line instead of being clicking these things. So I can have a long script that sets all the UI in the way that I like it. And then if I go back to Napari, now I can see them all together. Okay, and then I can start playing by hand as well. For example, I can just remove these layers for a moment. I can go to my blue layer. I can try to say, no, you know what? I want it to look like this. I'm exaggerating just so we obviously see the difference. And then if I put the other ones, then now my image looks, completely different. And this is the way in which you might have to explore very complicated data sets, right? So then you can start to play with the image view until it looks more or less as you want, and you can export that if you want as well. So this is just to show you that this is usually in most of the cases what you're going to be doing as an analyst is that you're going to load your data as a NumPy array. You're going to process it as it's just mathematics, right? You're going to do a lot of different operations, and at the very end, you are going to put it in the viewer to be able to explore it, or you are going to then quantify and so on and so forth. All right. So you usually start in an image-based fashion or even a small region of interest, right? Because you want to show that your pipeline is sort of doing what you expect. And once you have it, then you start running it automatically on a batch of images. Now, usually what you want is that you want to have some kind of evaluation mechanism, right? So you run it through, let's say, 20 images, maybe 20 images you can still evaluate by eye. But if not, at some point, you need to have some kind of automated evaluation, then you know that things are roughly working as you expect. So there is different possibilities. So what we tend to do a lot at the facility is that we have an image analysis server. It's a Windows server. And then in there is where we're going to put the heavy calculations. And that also gives me a lot of control. There was a question in the chat about the environments, because what I do is that I create an environment that I know it works on my server, right? So then I don't have to share the environment with you anymore. I just share the user access to the machine. So I give you the right username and password. We install everything together. We know it works, you know, in a Zoom call or something like that. And then you are running it and I have complete control because it's my machine. Another thing that you can do is that you can run it in clusters. You also know that, for example, even the Jupyter notebooks can be run on clusters and everything. So there is a lot of work running on clusters and it's possible. We haven't invested too much time on that, but I know people that have run it in both in CPUs and GPUs clusters. And now there is interesting possibilities here in Gothenburg area as well. You know, there is a new GPU cluster that they have invested in Chalmers a lot. So we are trying to talk to them because they don't have so many projects which are based on imaging. So we are interested in trying to use that to sneak in some time and just test stuff. So, but I have not done GPU clusters yet. All right, any other questions so far or? No? All right. So now let me just close this thing because I suspect that if I have many open, my computer is also not gonna like it, which I can already see that I cannot close them. Zoom and oh yes. Okay, so what I'm going to do is that I'm gonna run the segmentation without Napari. So what I wanted to show at the beginning of the book is that again, I do not use Napari like all the time, only for exploration, right? So there is no meaning for me to always be having an Napari viewer open if I'm not going to use one of the functionality that comes from Napari. So for example, here, what I do is that let's run it from here. This is gonna break, obviously, sorry for that, because there is no Napari viewer open. So let me just cancel that operation. Yeah, I know. But for example, here, what I can do is that I can, if Rise did actually load, yes, it did load. So you see here, for example, I can start working with my data as if it's just a numeric array, right? And then I can use things as matplotlib to look at it. So what I have been showing you here, I'm gonna close this guy down for the moment, is I have a classical image that is used for training in many fields, which is a Rise, and we're gonna try to segment this, okay? So the question for the people that are here, which is a bit easier to interact than in the chat, is this easy to segment or not? What do you think? Do you think that we can just do that super quickly, or there is any complication that you might expect from this image? You have seen it, so you have to close.
I would say that compared to
yes, it's extremely easy, but the question is, do you see already something which is very common in microscopy, which is actually going to be annoying at the beginning in this image? Yeah, go. Yeah, so
someone in the chat said background subtraction.
Yeah, exactly. So there is different methods there. I like your question mark about top hat. Yeah. So there is many different methods that we can try to use to do this. So the problem is, I'm going to show you first what is classical thresholding. So in classical thresholding is basically based on the analysis of the image histogram. Okay, all the algorithms, what are going to do is that they initially are going to build a histogram of all the counts that are available in that particular image. Now, please. Okay, there we go. So then what you see here is that in the ideal image, we should just have two populations, right? Background and objects. Right now, it's a little bit more complex than that. And the reason for that is what people are saying already, which is that this is an homogeneous background. So if I look at this histogram and I say, you know what, maybe the division should be there at 100. You know, I'm assuming that there should be two populations, right? So I'm gonna take that peak at 100. I'm going to divide exactly there and see what happens. And then if I do that and I look at my segmented image, then it looks like that. And the reason for that is because of the unhomogeneous background. And it doesn't matter how much I try to move my threshold. If I move it to 150, then at some point it works for some area, but not for the other area. Okay. So as it was described before, and this is just again to show you where you can find this type of functions in Python if you are just starting. So for example, skimage has a lot of filters. And in that filter, I'm gonna actually first load the Otsu thresholding. This is what I told you. That's the function that is used to analyze your histogram and find that value that you should be using for the threshold. So then I can say, hey, please find me the threshold value based on Otsu. So I can print that value and then I can just implement it, right? So Otsu says 131. Otsu is quite good, but not perfect. So then the question is, okay, this, if we don't do anything else, will not be ideal, all right? So then for that one, we're gonna do to the next one, which is this idea of filtering, which was more or less what was said before. At some point, somebody mentioned top hat. So what I show is that this is the original image, which has an homogeneous background, sorry for that. Then this is just the histogram that I showed you before. And what we are going to do is that I'm going to import now the Gaussian filter. And the Gaussian filter is basically like a blurring. The reason why we like it is because it's symmetrical, right? So it's like a symmetric blurring effect. And the interesting thing, you can play in this book yourself later on. Here is the sigma of that threshold. So sorry, of that Gaussian. So larger the sigma, more blurring, smaller the sigmas, less blurring. And then the interesting thing is that if I use a large blurring, I lose my rise because the sigma or the size of my Gaussian is larger than the size of my rise. Okay. Now this is the same as if you would be working in Fourier transforms. You can do exactly the same idea. And there's a lot of different filtering methods that you can use based on the fact that my background has a completely different spatial dimension than my objects. So that's a classical trick. So we can extract the background. Once we do that, we can subtract that background from my array, right? And then we get a flat illuminated image. And then from there, I can again call my friend Otsu. Please give me the threshold value. I can use it. And then we get much better results. And then just to show you, there's a small comparison at the very end between what we got in the original image versus what we got in the filtered image. All right. After that, I also want to show you that I love pandas. So all of this stuff is compatible with pandas. You know, down Excel love pandas. So just as a side joke, really. But the good thing is that now, if you use Seiki image, you can actually measure properties based on that segmentation, and all of that can be easily imported into a pandas data frame, and then you can do all your data mining afterwards in there, which is a lot easier, right? So again, this is just to show you the functions so you can play with them before. What we first do is that for each rise during the segmentation process, each object has the same value because it's just object versus background. Then what I have to do is that each rise has to have a unique identifier, right? It has to be rise 1, 2, 3, 4, 5 until 50 or whatever. That's what we did in here. So each rise has a unique identifier. And then finally, I will say, okay, I want to measure the area, the eccentricity, and the intensity. And then you can use this region props function from skimage to do that. So you say, what is my label image? What is my intensity image? And what properties I want? And then all of that table can be given to pandas, and it becomes a pandas data frame. And then this is just to show some very, very basic plotting. Okay. So I know that this is just to basically show you what is in there so you can play later on. So I'm gonna stop my share for the moment and go back to the chat. I don't see anything new there. Then the only other thing that I can do before we go, I'm gonna share my screen one more time. Go back to here. And then, so the other thing that I have is also in the PDF that is there in that website is a long list of resources. So you have links to many different things. And the one that I say, if you like the feeling that you get in Fiji or ImageJ, and you want to reproduce it in Python, and you don't like coding at all, then what you can try to do is that you can try to use the plugins that are made for Napari. So what that's going to allow you to do is to use a lot of image to image transformations. So a lot of filtering steps, a lot of these things you can do directly in the viewer. But also there's a lot of very exciting stuff where, because Python is behind and Pandas is behind, you can even start doing some clustering and data processing of the output tables, everything within Napari, okay, which would be very, very difficult to manage in Fiji. Then there is some other different websites, so just please go in there. And then, of course, I just want to quickly show all my colleagues, just to let you know who we are. If you are interested in our services, you can please just go to that website. We receive funding from different, from different organizations, of course, and just to let everybody know that we are recruiting, especially in this concept of smart microscopy. So if this is something that is interesting to you, we're going to open a position soon. So just send me a message afterwards, reach out, and then we can talk about. So with that, I end for today.
All right. Thank you. Very nice talk. Yeah, I mean, we have a few minutes left. If anybody has any questions in the chat, right? Alexander, right?
Yeah, the link to the resources, I'm going to put it one more time. Absolutely, no problem.
I can answer it.
Yes.
Like the images you analyze,
yes.
Are they only generated at CCI, or do you get like sent images from other microscopes from around the world?
Yeah, so the question again is, if we do, basically, if we provide image analysis services to people that come with their own images. And the answer is that yes, we do, but we do give priority to people that work with us, right? So of course, if I have 20 different projects that want service and 10 of those come from the facility, I will service those first before I service the other ones. But we do offer image analysis to the outside world. Also, one thing that we tend to do if we are way too busy is that we have this model in which is a little bit more sort of collaborative. So it will involve much more of your time than my time. But what we can do is that, for example, we can have a meeting of 1 hour where I show you what is the way I will do the analysis and what are the tools that you should use and learn. And then we define a task that you think you can do without me. And I send you on your way. You try that task, and then you come back to me either with questions or when you have managed. And then we go to the next step and the next step, right? And I think that also a lot of PhD students really like that model because they want to learn everything. If you are more like a postdoc and it results very quickly and your data is humongous, you don't like that. So it depends a little bit where you are coming from, okay?
Nobody, no more questions?
There is a package to interact with image from Python.
Sorry, could you repeat the?
There is a package to Python to interact with image from Python.
Yes, that is correct. Yes, yeah. Yes, for very specific functions that are in ImageJ, we do use it. But if I can do it from a library in Python, then it's easier, to be honest, right? But the thing is that there are things in Fiji that are very, very, very interesting. So then you can, I know that even recently there was a new article that came out with even better integration. So I can, if you're interested, I can try to find it out and send it your way. So now there's even better ways of calling Fiji from within Python, okay? The other thing that I would recommend is that I really use Fiji almost every day. So I think that community is great, nothing against it. The only thing is that if your data sets start to be like really 3D or really big or so on, that viewer is not as responsive as Napari. So you of course could go into the big data viewer of Fiji, which has a lot of stuff as well, which is quite impressive. You can use it. But there is like a small niche for different things depending on your application. Okay. And from my point of view, I think the most exciting is this collaboration with other communities that do, for example, the pure, bioinformatics guys that that handle pandas data frames all the time and do all of this magic stuff. They would be kind of very happy if everything stays there, right? So it's a little bit of what do you need for your project. If you need data mining, I think this is a good way to go as well.
Yeah, one more comment.
Yes, so yeah, some very interesting. Yes, so the integration with pandas data frames is very cool. And then also there is a lot of people that are doing this sort of kind of already clustering with a UI in the NumPy viewer. So you could have a set of, let's say, 100 images in the NumPy viewer that then you analyze and you create a large data table. That data table, it's known to the NumPy viewer. And then you can take your different rows and for example, a k-means with some kind of UMAP, you know, these sort of things you can already do in the viewer. And it's also responsive. So if you see a cloud of points in that UMAP, you can kind of circle them and you can see what it corresponds to the image. So there is a lot of interesting efforts going there, extensions of the NumPy. All right. So I'm gonna stay around a few more minutes. So if somebody has a question, just let me know. And if not, thank you very much for everything.
Maybe people can email you.
Absolutely. Yes.
Okay. All right, great. I guess we'll round it off there then. And see you next time.
See you next time. Yes.